I think we'll go ahead and get started. Certainly my pleasure to welcome Dr. Mahendra Rao to IBB and to give our bioengineering seminar this morning. Just a little bit of background, hopefully not too much. He actually is an MD, Ph.D. with his MD degree from Bombay, Ph.D. from Caltech. I think you probably all have heard of Caltech at some point. But did a couple postdocs, was on the faculty at the University of Utah, but ended up in the industrial world. And when you look at his CV, he has a variety of titles, but basically he's with the Invitrogen part of Life Technologies and Vice President for Regenerative Medicine or something like that. Is that correct?
That's correct.
So he's certainly been an important leader in this whole area of the translation of benchtop stem cell biology to patients, to clinical therapies, to commercialization. So it's great that you could be with us. I know your schedule is always very tight, and so he flew in this morning. He's flying home late tonight, but we have him for the day, and we're very fortunate to have him. So Mahendra, welcome very much.
So it's a pleasure to be here. I've actually never been to Georgia Tech before, but I have been to Emory several times, and I had a series of collaborations with different groups there, but I won't talk about those. So it's always nice to come back and especially meet some of my colleagues from Athens, Georgia, whom we worked with as well. So I'm going to start off my talk by giving you an apology, right? I'm going to tell you first, confess that I'm not a bioengineer and I've had no bioengineering training whatsoever. On the other hand, I've dabbled in bioengineering all throughout my graduate school and postdoc and faculty days. At one extreme, I worked on space biology and looking at zero gravity microculture systems. On the other end, I looked at power requirements for transmitting electrophysiology, multi-unit recordings to using wireless-based multi-coding techniques. However, it also means that since I didn't know what I was doing, most of those things did not work, right? And so some thesis of my talk today is going to be the advice I give to a lot of my students, right? Is that I think a lot of us, when we do bioengineering as a degree or as a career, start off by trying to solve the most difficult problems first. And I think it's really important to learn to walk before you run, and so you'll see that the whole experiment and what I describe in the next set of talks is going to be about how to do that. And then secondly, one difference between what bioengineering students do and what my science students in biology do is that we don't think of solving the whole problem. Engineers like to focus. So they focus on one specific piece of the problem, and once they've solved it, they've declared it as a success. But that success doesn't necessarily translate it to being used because it's not part of a general solution. So what you're going to see in my talk today is taking very simple solutions and applying them in a comprehensive way, and hopefully you'll find or you'll see why this thesis might be important. Okay. So I'm going to tell you two stories about work that we've been doing. One is going to be on mesenchymal stem cells, and the other is going to be on neural stem cells or embryonic stem cells, and using them as a source to derive neural-related stuff. Okay. So for those of you who don't know anything about mesenchymal stem cells, they're an adult type of stem cell, and if you look along the side here, which is outlined in red, you can get mesenchymal stem cell-like cells from a variety of sources. So you have bone marrow, adipose tissue, umbilical cord, amniotic fluid, skeletal muscle, etc. There's also a subclass of mesenchymal-like stem cells which the inventors or discoverers have claimed are different because they have properties in addition to what MSC have, but they're overall similar, and those include things like MAPC, MPLC, Miami cells, very small, ultra-small, pluripotent stem cells from adult tissue, etc. Okay. And then there's the idea that you can use other sources of cells, such as embryonic stem cells, to give you mesenchymal cells. There's a process of transdifferentiation where you may be able to take cells and make them like mesenchymal cells. And then the two important things that we learned from the field were that there are one class of therapy where people think about using mesenchymal cells as undifferentiated cells, and that includes things like sort of basic biology, cancer biology, and one subclass of cell therapy where we're thinking of graft versus host disease, etc. And then there's a process where you can say, I'm using mesenchymal stem cells as a source of differentiated cells. And what you see here is a whole series of differentiated phenotypes that have been shown to be derived from mesenchymal cells, both in vitro and in vivo. And so those include tenocytes, chondrocytes, osteocytes, adipocytes, myoblasts, etc. So you can imagine that people working with cartilage may want to take mesenchymal cells, pre-differentiate them into cartilage, and use them for therapy. So keep all of this in mind, because this is the problem, right? How do you devise a process so that you can scale up culture of these cells and do it in a complete system that can be applicable to the largest number of people using stem cells? Okay. And so the story I'm going to tell you in the next few slides is looking at that kind of solution. Okay. Okay. So when I first started thinking about this problem, you can imagine thinking about this problem by going from the reverse end. So taking a medical background and saying, what is the kind of therapy people are thinking with mesenchymal cells, and using that to work backwards in terms of what is your scalable issue or problem in terms of manufacturing cells. How many cells will I need, and for how many patients? Right? And so broadly, we can divide that into saying that mesenchymal stem cell therapy can be broadly classified into two types. One is where it's autologous or a personalized medicine type of therapy, and the other is allogeneic. And for me, it doesn't matter what that means in terms of regulatory authorities, at least for this talk, but what it means is if it's personalized, then I'm taking a small number of cells and I'm amplifying them for a limited amount of time. If it's allogenic, then I'm going to be doing a large-scale manufacture where I'm treating 10,000 patients with the same source of cells, so I have a much bigger scalable issue problem. Right? So personalized medicine might require a different solution. Large-scale manufacture will require a different class of solution. And I need to worry about both of those sorts of solutions. Okay? So right away, that gave me a very simple answer. If I want to solve this problem, I need a complete system, and I need a system which might be viable for small number amplification, and I need a system which would be viable for a really truly large number amplification of cells. And I need a system which is for growing the cells or expanding them, and I need a system for differentiating them. Okay. So that seemed fairly straightforward. Now at least I had a handle around what kind of problem I was going to try and solve. And since I needed a complete system, I thought, let's see what we can start with first. And here, it was really quite useful for me. We talked to a lot of companies which are already working towards therapy here, and we asked them what their process was so that I could identify all the things that I needed to solve to get to the complete system. And the answer was, and this is taken or borrowed from a company called Athersys, which is based out of Cleveland, Ohio, and they work on a cell which was first identified by Catherine Raphael, and they have a process for their INDs that they're going to perform. And so you can, and I'm just using it as an example, it really doesn't matter. I could have used it from some other company such as Osiris, right? So it's isolate material, you take this, you process it, hopefully you want to do it in a closed system. They needed to expand it, and I'll tell you a little bit about the kinds of trials they were running, so the numbers that they needed. Then ultimately, after you've expanded the cells, you need to concentrate them into your end product at some fashion. You need to create a master bank. You need to take that and make a working bank from which you're going to get lots and lots of clinical doses. And then you need to ship it to the end user, and then you need to thaw it and do some kind of qualification tests that are required by the FDA, and then you need to administer the cells. Okay. So if I can break it down into these steps, then for each of these steps, I need a solution. And that solution, in my mind, requires bioengineering solutions because I need a device, I need media, I need storage solutions, I need temperature controls, I need sensors, etc., in terms of being able to do it. But I need to make sure that they all work together. Okay. And I knew which number I needed based on their protocol and trials and the number of patients that they wanted. And so we won't go through all of this, but suffice to say they needed much larger numbers, and they needed to treat tens of thousands of patients once the trial was successful. But for the initial trial and manufacture, they needed a relatively small number because they were thinking about 50 patients at a time, and they didn't have a bigger issue on shipping the cells to multiple sites. So we'll skip that. And they were using undifferentiated cells. Okay. So I'm going to focus then and just tell you that this part was pretty straightforward to address. It's not anything novel, so I'm not going to talk about it for today. It was pretty much what we've been doing with tissue processing and tissue sourcing or making cell lines, so that part could be solved pretty easily. Then we had to worry about tissue processing and figure out what would be a system which would work together, and then see whether we could have it in a closed system and what would be there, and then figure out what would be the quality control that needed to be done. I'm going to quickly go through all of this, and you don't need to worry too much about the details on the slide, because as I pointed out, I'm talking about simple things and really putting them all together rather than something which is technically really novel or completely challenging. Okay. So the first thing that became clear was that we needed to figure out and define a media, and that media need to be able to work with multiple cell types, because there were multiple sources of isolation, but they were all related cell types. And then I needed to figure out a media which would work at different scale. And this is an important issue that people need to worry about. Now, I could have started by doing this empirically, but then that's a problem, because I'll empirically do it, but I won't quite understand the basis of why that media worked. And every time I have to vary it for a new cell type, the effort is going to be about the same. So is there a way I can do this in some sort of generalized process, or at least identify the basic mechanisms by which one can design a good media? If I can do that, then I could solve the problem for most of these cell types. And I'll tell you a little bit about what I did. And that's all summarized in this one slide. Okay. So what I decided to do was to say, let's take advantage of the genomic revolution that we already have, and the way that we have technology in terms of being able to look at gene expression profiling. And what we'll do is take software and analyze mesenchymal cells. Okay. And what we'll do by this analysis is look for all signaling pathways that are active in mesenchymal stem cells when they are being grown and expanded, right? Because there was a standard sort of sera containing media which could be used to grow and expand the cells. Once I knew that, I could also check which growth factor receptors are present on the surface of these cells based on this sort of gene expression analysis. I could verify that by proteomics, and then I could go back and check whether those corresponding growth factors had an effect on these cells. Since I could compare across multiple cell types, and I could compare at multiple stages in the differentiation process versus proliferation process, I should be able to identify the key pathways that are important for keeping them proliferating. If I can do it in a relatively cheap and quick way, then I have a way to design media which may be relatively generalized. The answer turned out to be yes, we could do this, and this was done within the Invitrogen environment by a research group. So this is the research part, not the manufacturing and production or the commercial part of Invitrogen. And what we were able to identify, and again, we identified several critical pathways. The three that seemed to be the most important were inhibition of TGF-beta signaling, activation of PDGF signaling, and activation of FGF pathways. We could look at alternatives, such as VEGF might work through the same PDGF pathway, and we had a lot of data on that front. And we also knew what were the additional things which could synergize with these. But these were the three that we chose because they had the biggest bang for the buck when we did some confirmatory tests. Right? So now we knew that we had a generalized system. These three pathways were important for most of the mesenchymal cell populations or mesenchymal stem cell-like populations, and we should be able to define a xeno-free or a serum-free media relatively quickly. So the next part of it was pretty straightforward, so I won't talk about it except to say, yes, we could do it, we could validate it, we could test it, and the first media that I could produce from all of this stuff was a serum-free media. So we had found an alternative to serum. We could do it in a relatively short period of time, and this media worked with rat, mouse, and human mesenchymal cells, and it worked with bone marrow-derived cells and worked with cord blood-derived mesenchymal cells and worked with cord tissue-derived cells as well. Okay. So that was first step. However, this is not enough for the regulatory authorities. You really don't need just a serum-free media. You also want it to be xeno-free. And you don't just want it to be a media solution. Remember I said you have to start with having a whole package that works together. So we had to figure out whether you need a substrate, what would be the passaging enzymes you would need, whether you would need to alter anything within those components that might be important in terms of sourcing, what would be reasonable for manufacturing processes. So it turned out that we needed a substrate. That substrate had to be of humanized protein, so we were able to identify something that looked at that. We turned out that a simple change from glutamine to glutamax, which doesn't release as much ammonia into the system, was important in growing the cells. We found that a recombinant enzyme like triple could work in passaging the cells. We also found acutase worked, and so we had a list of enzymes which could be used for passaging cells, and also a mechanical method of being able to do it. And we could show that all of these things work together. So now we had a combined system which included media, substrate, passaging system, as well as a cryopreservation system, which we could use for expanding the cells. Okay. Because whenever you do all of this, and this is a really important thing for all of us to keep in mind, is you always have to test against some standard. It's not just that it works. The question is, does it work in a cost-effective way? And how will people compare it in terms of whatever is commercially available otherwise, in terms of defining success, right? And so this is just a series of tests that we did, and I won't go through them in a whole lot of detail, but we looked, except for two. Okay, one important test for us, because this is sort of a release criteria when you're making your end product cells, is, are the cells functionally good, right? You grow them, you've changed the media, you need to make sure that the cells are functionally appropriate. And so that's what you're looking at in the right-hand side here, is that the cells can differentiate into the three major lineages that are a de facto standard. What's important really here is to consider chondrocytes, because that's the property that's lost with mesenchymal cells early. So that was a good sensitive indicator. We also wanted to look at growth rates and other characteristics, and that's what you found, that the cells grew at least as well. In this case, it's actually better than cells grown in serum, and they didn't undergo senescence that early. You could also, you needed to measure any kind of change when you've changed conditions, when you're growing cells on a large scale, and you have to figure out a way to monitor that. And so we looked back at a whole gene expression analysis, and we just used a simple correlation coefficient, right? If two cells are similar, then the whole pattern of gene expression that you see should be very similar. If they are different, you should be able to distinguish between them. And because you can use an R-squared correlation coefficient to look at that, you can use that as a number to define how closely related your cells are. And we've shown by other experiments that we published that this is a very sensitive method, and an R-squared correlation coefficient of 0.99 really suggests that the samples are identical. If you have two related cells or two different isolations of the same population, you're generally in the 0.9697 range. If you culture the cells and you grow them for 10 passages, that's what you will see between the first and 10th passage. That's the overall correlation coefficient. And in this case, multiple isolates, or you grow them in serum-containing media, or you grow them in the serum-free media, that's the overall correlation coefficient. So what it suggests is that your media is substituting for serum. It's not changing the cells in any significant way, and on an overall global gene expression way, in an unbiased way that you can look at it, and the functional criteria is confirming that, right? And the growth rates are good. So overall, this tells you that you have a good media and that it works reasonably well. I'll skip this. This is in vivo work. This is required for regulatory requirements if you build something. So I'll skip that, except to say that it all confirmed what I just said. So I'll skip that. So the next thing that we had was, now, remember I said, don't run before you can walk, right? So that was my first step, right? We had a serum-free media. Now I had a control, and I had tests by which I could compare if I wanted to improve this. So what was the improvement that people wanted, right, for mesenchymal stem cells? And the thing that they wanted was not just a serum-free media, they wanted a xeno-free media, because animal components require a lot more regulatory hurdles that you have to pass. But now I had a process, right? I have media, I know all the components in that media, I know which ones come from an animal source, so I can go back and check all of those, and I can see if I can swap them out with either a recombinant source or a human protein source, right, to make it xeno-free. And I won't go through the whole story because it was pretty straightforward in terms of being able to do it. I'll just tell you the problems that we had in terms of doing it. But we came up with a xeno-free media as well. And what I want to emphasize here is not that we came up with the media, but this relative speed with which one can do that if you deduce things from first principles and you start by making sure that you identify the basic science and the pathways and have an understanding of what went into designing that media. So we made a xeno-free media, and when we did it, we had two problems. Okay? The first problem was that all recombinant proteins didn't work as effectively as purified proteins from human sources. So we don't have a fully defined media. It certainly works. The other problem that we had when we did this development of this media was how long the experiments take. And this is something I want to emphasize here. It's also going to be important for the second half of my talk as well, is that to test the quality of your cells, if I use a short-term passaging experiment, I can see very nice quality results with a whole variety of things that I discarded. So my cutoff is that they have to grow for 8 passages, right? With human cells, passaging is every 5 to 7 days. So doing this experiment for 8 passages and then differentiating, which takes me 3 more weeks, is 56 days plus 21 days. That's a lot of time, especially when you work for a company which is reporting results every quarter. Okay. So being on the research side, I felt the heat. So that was a big problem. But I have no solution for that. So this is what I'm going to recommend to all of you. It's easy to say success when you're looking on a short-term rate. I actually have 5 different other growth factor cocktails that will work really well for 3 or 4 passages, but will fail by the 7th or 8th passage, right? And that's a really important thing to keep in mind, because it's easy to get fooled. And that was even more important for embryonic stem cells when we looked at that. Okay. So now, the next obvious thing, of course, was to say, whenever you change a media, does it work with all the other components that you've put together? Because that's one thing you learn when you do all of these things, is it's a complete package. You change one thing, there might be resultant changes in other things as well. But in this case, we could show all of that, and we could show that there was a complete package and that worked together as well. And we were lucky there that the same components that worked with serum-free worked with xeno-free. These were all the same tests that I showed, and the only thing I want to highlight here is that the growth rates with xeno-free were not much better than that with serum. So they met the standard, but they weren't better, unlike serum-free media was. But that's generally a common price that people pay whenever you go to xeno-free and for large-scale manufacture. But that's something to keep in mind. These are all the additional rigorous tests, because now you need to worry a lot more whenever you're making clinical grade stuff. Is the karyotype maintained? We did SNP analysis to show that the cells behave the same way. You looked at multiple lots. You look to see, markers by FACS analysis. You look to see which test would be the right test for reproducible lots in terms of being able to do it. But these were all generally relatively standard off-the-shelf things which we just simply had to adapt. Okay, but we needed to adapt them so that we had a complete process of being able to do this. Okay. So I'll skip all of this in the interest of time, and I'll re-emphasize this because I'm a strong believer in doing genome-wide sort of gene expression analysis as a good sensor, for being able to do it. Not necessarily the discovery for which individual gene may be changed, but just as an overall state of the cells, it turns out to be a very good sensor. Prices have come down really significantly for being able to do these sorts of tests, and I can do the same sort of 30,000, 35,000 gene expression profile that it would cost me to do by PCR for maybe 50 genes or 25 genes. Okay. And what you see below here is just a hierarchical clustering, just to show how sensitive this can be in terms of being able to differentiate cells. Okay. So the next question then is, you've got a media. That media works. You've shown it works as a complete package, and it answers one part of the differentiation process, the development process. Will it work when you're isolating the cells? Can you isolate in this media? If you can't, then you have a problem, because now you have a separate development protocol that you need to worry about. And if you can, then you've saved yourself some time. So the good answer here was that for bone marrow isolation, the xeno-free component worked pretty well. And so we did this with multiple groups, and so you can do a fresh isolation in this condition, okay, with this complete package. Okay. I'll skip the comparisons. The reason why I have the comparisons here is that you have to be quite rigorous in testing against standards, right, or whatever is there, to be able to make sure whether you will have a basis for improvement. But even on a development scale, what will be your comparator if you have to build something better, right? What will be your cutoff, and how will you be able to know whether that's true? And it's not easy for me to say I can build something which will be better than anything unless I know that the cells are actually capable of behaving better, right? I don't want to push the cell cycle too much, because I can have the cells grow faster, because if that's going to cause them to become karyotypically abnormal, then I have a problem. So I need to know how much I can step on the accelerator first right before I do that. So it's always important to check a wide variety of different conditions in which the cells grow to know how you're going to set up your test experiment. Overall, the take home here was that the cells didn't grow as fast as serum-free media, but the difference was about 10%, and that 10% was my range, and I didn't think I could use that range as a good sensitive test as a readout to be able to develop a better system. And so it seemed like in the time period that I had. So this seemed like the best thing I could do with the time period I had. So now that I had sort of one leg to stand on, so I knew that I had a solution for the first two parts of the isolation of the cells and to expand the cells, and I had a solution to qualify them. And I didn't show you all the data, but just assume that I do, because we tested it. We have a storage solution as well. It's a storage solution which takes DMSO, but works seamlessly with the cells grown in this particular media. And we have different ways of monitoring the cells. And you can ignore the rest of the slide picture. It's just basically to give you an idea of the various kinds of tests that we used and the options that we had to be able to do it. So are we done? That's the next important question to ask oneself. And the answer is no. You're not yet done, right, in terms of being able to say that you have succeeded in providing a complete solution. You can isolate, you can expand, but I haven't shown you what is the size and the number we can expand in, and I haven't shown you that's all compatible with closed systems as well in terms of being able to do it. And those are the next sort of important things to be able to do. And I need to show that I have the cells in a sufficient state that I can take these cells and I can do all the appropriate testing that's needed. I just showed you some ways that we can do the testing. So I'll skip that part and I just focus on some of the closed systems that are there and what we need to do to evaluate them and what issues there are with size. And so I won't go through all the details, but I'll just highlight some of these things and say we've been working with different collaborators here to trying to figure out that we can have closed systems and we can walk from one closed system to another through the whole process. And this is actually pretty kludgy, right? It's not like we have a nice single device that works all the way together where I can just feed in a starter cell population or the bone marrow aspirate and then get manufactured cells at the other end. But what we can do is do the following. We can pick from what people have done with closed systems for harvesting bone marrow or blood. The bags have been worked out, the size of the bags has been worked out, the sterility of the loops and the plungers and the valves has been worked out, and I can just piggyback on that. So I can take bone marrow aspirate and collect in the same kind of bags. There's been methods worked out to harvest mononuclear fractions from bone marrow or from blood in a closed system. There are actually 26 such devices that I'm aware of to be able to do that. So we've tested some of those closed systems with media and components, etc., to make sure that we can link these two pieces together. We've then taken that and looked at various closed culture systems. And we've done more than this one, but this is a standard sort of cassette closed culture system which has fluid flow in it to be able to expand the cells. And so this is towards the more personalized medicine. This is certainly not at a large scale. And then we've looked at seeing whether there's a closed system in which we can harvest and collect cells and how we can deliver them, right? So these are harvested, collected cells from which we've gotten rid of trypsin, and then we have a delivery device which is much more like you would have when you infuse cells IV, and there are clinically approved systems for being able to do that. And then how can you store? Can you store in a bag in which you can thaw them out so that then you can ship it back to the site and where you can deliver them? So we tried to look to see whether we could move a fresh harvested MSC population and take them through this entire system, and would that work, and would it work for clinical trials? And I don't know if you can see this, I apologize for this, but it's a group at the Ruhr University in Germany in which we've been looking at this and looking at all the data in terms of being able to use a closed system to do it. So the obvious next step will be to take these pieces and try and see if we can put all of this together. Okay. The next piece then is, can we do it on a really large scale? And again, many things have been worked out on large scale, and I'm going to just highlight a couple of strategies that we figure out here. One thing that was really important was, can we devise a way to ship bulk media for a really large scale manufacture? it's a lot of fluid, and it's a lot of liters that you need to really do. You manufacture media in one place. If you really have to do a commercial scale production, we have to figure out a way to do this. Turned out that this had been solved for all the bioproduction needs that are being done with biopharmaceuticals. And the thing that we chose is this thing called the advanced granulation technology. It's not lyophilization, because with lyophilization, resuspension is quite difficult. With granulation, getting the substances into solution and stability seem to be quite good. So we know how to do this. We just checked whether we can take our media and do the same thing, and the answer was yes. And the next question was, can we get the right size bags, and can we make a closed system in which we can take this media, take the right film, so that you can really now grow in bulk culture. And I just wanted to give you examples of two of these sort of off-the-shelf commercial systems that are available. One of them is from GE, which is this sort of wave bag technology. The other is sort of a modified intermediate scale technology that you can do. And we just checked to see whether you can do pre-filled systems and whether you can actually just fill this with granulated media so that you can add water and cells and get a fresh pack that you can use for all of these storage requirements and for manufacturing requirements on a large scale. And that's an example of doing that with AGT. So you design your bag in the right way so that you've got the media loaded in one side. This is your culture system. There's a seal which you break, and then you add water to this so that you solubilize your media, and then you break this seal, you add cells here, and you have a culture device that goes into a bag or a shaker or wherever you want appropriately, and you have appropriate connectors to be able to do this. There are also single-use bioreactors which we've tested. And the importance here is we're taking advantage of existing development and adapting or tweaking rather than trying to solve a problem from scratch or looking at a completely unique solution in terms of being able to do it. And importantly, we are testing that there's a closed system that we can deliver in time to have success. Okay, so I want to try and emphasize all of that in terms of being able to do this. So these sort of universal bag solutions actually exist. There are companies which sell them, so it's just a matter of adapting existing things to be able to do this. Okay. The last piece I want to tell you about in terms of a technology which I think is going to be important overall is how do you select cells? So this is sort of a fundamental problem that's been facing us, not with just growing undifferentiated MSC, but certainly a problem with other stem cells as well as when you want to differentiate cells. Because one weakness with our technology right now for differentiating cells is that there's nothing that we can do which gives us 100% of one kind of cell. So what we have to do is get some efficiency and then purify if we need a purified population of differentiated cells. So we've looked at a whole variety of different technologies in terms of being able to do that. And again, our goal is a successful complete system which works with all the media, reagents, bags, etc. that we put together, right, and the closed systems that I just described, which will allow you to do it on a clinical scale relatively quickly because people are moving towards therapy relatively quickly, right? And so we looked to see what kinds of technologies were available which were safe and relatively reliable and which could be used at a GMP scale and which could be GMP-able, which unfortunately for us right now, FACS is not. Okay? And there were two bead technologies that seemed to fulfill that criteria. One of them, luckily for me, came from the company I work for, which is Dynal Beads. There's another company which manufactures another magnetic-based technology, which is Miltenyi. The difference is the Dynal beads are much larger. They're much larger than the size of the cell. They don't get ingested. Miltenyi beads are much smaller than the size of a cell. So there's an advantage of working with them, and the disadvantage is that they get taken up by the cell. So there's a pros and cons to that. But both are clinically approved sets of beads which have been used for purifying or isolating cells for clinical trials. Okay, so clearly a clinical grade product existed, and what we needed to test was whether we could use it in our closed sort of system to be able to do any kind of selection should we need it in the future. Okay? And the take-home answer is yes, we have a prototype, but we haven't yet tested it and shown successful large-scale isolation using this system. Okay. However, we've been successful in using the system not to do positive selection, but to do negative selection. And negative selection becomes quite important, and I'm not going to show you this data again, so I just thought this might be a good time to show this, is that we can take dinyl beads, link them to SSEA4. SSEA4 is an antigen which is present on undifferentiated embryonic stem cells, and we can get rid of the undifferentiated cells during the process of differentiation so that you reduce the risk of teratomas. Okay. So depletion works. We can use clinical grade beads to be able to do it. Positive selection we haven't yet shown on a large scale, but we think that we have a process and a technology to be able to do that. Okay. So I'll just end this section and spend the next 10 or 15 minutes on looking at neural stem cells, just to highlight the sort of conceptual differences. So we can take a closed system, we can isolate the cells, we can look at the properties of the cells, and we can do the same sort of rigorous analysis that I just pointed out in being able to look at whole gene expression, morphology, ability to differentiate, ability to make different tissue, and we think that all of those things are now possible. Okay. I'll skip all of that and go straight, in the interest of time, to looking at neural stem cells and embryonic stem cells. So the only things I'm going to highlight here are the differences, or what is the additional scope of the problem as far as we are concerned. So hopefully I can go much faster. So the one important difference here between adult stem cells or mesenchymal stem cells and embryonic stem cells is that there's really no therapy with just undifferentiated cells. I just use them as a bank to get whatever kind of intermediate progenitor, precursor, or fully differentiated cell that I need. So that's one big problem that I need to keep in mind. It means I always need some selection process with currently available technology, and my processes are going to take time because I have to differentiate them. The second important piece is time. And time involved with multiple culture conditions, okay? Because what is optimal for growing cells is not optimal for differentiating them. So it means that my processes for growing embryonic stem cells or having a complete system are going to be multiple components or stages, and it will require that I solve multiple media problems along the way. Okay. And my scale-up issues are going to be different, because when I make a seed bank, it's going to be relatively small. When I make a master bank, it's still going to be relatively small, unlike what I had to do with mesenchymal cells, which are being used at an undifferentiated state, but I'll have to expand downstream in a different way. So my scales will be different at each of these stages as well. So keeping all of those in mind, I'll just quickly go through a lot of slides, and again, we'll just highlight some things on that front, and I'll skip this, except to point out that just like with MSC, we needed to make sure that whatever we did on the expansion side also worked on the derivation side. Okay, so we used the same strategy. We looked at which pathways and which growth factors might be important in terms of the self-renewal of the cells. We figured out which were the key signaling pathways and how we could use small molecules or growth factors to make a defined media. We used the same sort of slow step strategy in terms of being able to go with this. We started with a serum replacement. We went to a serum-free media, and in fact, part of the serum-free media development was done in collaboration with Brisagen, which is in Athens, Georgia, under the guidance of Alan Robbins, okay, who in fact you guys interact with. And we were able to show with the same rigorous criteria that you can get cells which can be maintained in an undifferentiated state. You don't need feeders. You can grow them without feeders in a growth factor cocktail. We did the same sort of experiment. Here we needed to worry about whether we needed them in a large scale for certain conditions, and we showed that these can be grown in suspension culture, and this was done in collaboration with Alan, and in fact this slide is from Alan. Okay. So just using this sort of mapping out process, we went through the same sort of strategy that we did before, and I'll just use this to summarize a lot of the data and just show you some of the controls in the next few slides. So we looked to see whether we could get these SOPs and the IVF clinic or the iPSC source, and that the derivation could be done in the same media that we were going to use to make all of these steps, and the answer was yes. Did we know we had to make sure that we could grow them in a small scale when we were making seed banks, or we were making a master bank in the same media, substrate, enzyme passaging conditions, plasticware that we were using? And the answer was that was true. So then we had to try and go to the next step. So here we decided that we wanted to introduce an important step in our normal manufacturing, because this was going to be too long, to see whether we could freeze and thaw the cells as part of our regular process with reasonable viability. And the answer was yes as well. And we had a media, and just like we said that for all of this we need xeno-free media, we had to make sure that all of that process would be true. So I'm just going to try and focus on these steps and say that this seemed to be like a generalized step that we need from ES cells. We needed to initiate differentiation, we needed to optimize that process, we needed to have some kind of selection protocols, and then we needed to have something on shipping. I won't tell you anything about shipping, and I won't tell you anything about selection and depletion protocols since I mentioned them already, and I'll just use one example, and that is how to make dopaminergic neurons using a large-scale process like this from ES cells, because that's a differentiated cell population. It's difficult to use, it's got multiple processes, and hopefully the problems that you have to solve are similar to that in any other cell type. Okay. So relatively quickly now with the next few slides then. So we looked at many different protocols. It turned out that there were maybe one standard protocol that we could use, and that was to make EBs. From EBs make neural stem cells. From neural stem cells make what looked like midbrain to finally get dopaminergic neurons. Okay? So we took that process and we said, can we now make it scalable? And the answer was yes. We could take xeno-free conditions, make embryoid bodies from them. Initially we did it with I6, but then we took a clinical grade line and derived it that way in the same media, and then we showed that we could grow it. We went through a step where you make what are called rosettes from those embryoid bodies. We harvested these rosettes, and the reason I've highlighted this is that this is a process we haven't still been able to automate in any efficient way. So this is a manual process. It's okay at this scale that we are doing it now, but this is going to be an issue, and it's a technical challenge that we haven't sorted out here. We take these rosettes and we harvest them, dissociate them, and get single cell suspensions. We grow them on an adherent substrate. We can also grow them in suspension. I won't show that data, and you get a pretty homogeneous pure population of neural stem cells. Those neural stem cells can be grown, and we know that after freezing and thawing them that we can differentiate them. Okay? This is just to show that we can do this all with clinical grade lines which have been derived in an IVF clinic under xeno-free conditions. We'll skip that slide. We then had to figure out how to make them midbrain, and again we took advantage of the literature and a standard strategy of being able to look at all of this. And again, I won't go through all of this. It turned out that there were two useful growth factors that we could use, FGF8 and sonic hedgehog, and if we exposed for a period of 10 days, we would get the neural stem cells to convert to a midbrain phenotype. That midbrain phenotype included neurons and support cells and undifferentiated cells, but it was a critical requirement before you got the right kind of dopaminergic neurons. Okay. I'll skip all of this. This is all verification using mouse models to show that the growth factors we'd identified were the right ones, because they also worked in mouse in vivo transgenics where we looked at knockouts or overexpression studies. Okay. So the end result was that we had a process by which we could start with a clinical grade line in clinical grade media, grow them up, freeze them at the master bank stage, get them to the neural stem cell stage, freeze at the neural stem cell stage if we needed to, and go through two additional steps where we could get about 10 to 30% of the cells being dopaminergic neurons of the right phenotype. We can't get more than that unless we take the cells and we use a purification protocol. And as I pointed out, we can deplete unwanted cells, but in a positive selection, we can't do it on a large scale. We can do it on a small scale, and it works for animal studies, etc., but it doesn't work on a large scale yet in our hands with sufficient efficiency. And I'll skip all of this. This is just the last shipping part. Can we freeze the cells and can we thaw them, and will they still survive, and will we have reasonable viability? The FDA has some specific cutoffs that we need for that, and the answer was yes. So at the stage that we want to transplant in a theoretically clinical environment, this would be the stage we'd want to ship to the hospital. This would be the stage that you thaw them out, and this is the stage you transplant. So that's a quick summary, that this is what we can do with these cells, and that we can get them in the appropriate stage, and this just highlights the stages at what we need to do this. Okay. This is just to show where we are with positive selection. Remember I said that we can't really do it at a large scale, but we can do it on a small scale. So we've been doing some testing, and it turns out that we can use two antibodies to be able to get positive selection. One of them is what you see here, that's PSA-NCAM. PSA-NCAM is not just expressed on dopaminergic neurons, it's actually expressed on postmitotic neurons, many kinds, but it certainly enriches away from glia and other support cells and stem cells which are present in the culture. So you can get a significant enrichment of your dopaminergic neuron population along with other neurons if you use that marker. if you combine it with one additional marker, and that's low affinity NGF receptor, you can get a purity of about 80% in terms of being able to do it. We can't combine it by using 2 antibodies at the same time, but we can combine it by doing a sequential purification process. The viability for that sequential process is not such that we can use it at a large scale. It's fine for using on a small scale to do animal studies. Okay. So I'll skip all of the depletion. And this is just a pretty picture to show we can get relatively pure isolation. Okay. So are we done here? So like MSC, there are issues with the manufacturing protocol, but with embryonic stem cells, we have one other important issue, and which I just want to highlight in one slide, since I have a couple of minutes, and then I'll stop, which is what are the tests you'll do, and are negative results reasonable? Okay. And I want to emphasize this, even though it's not truly a bioengineering problem, but it's a problem which I think lends itself to a bioengineering solution. Okay. And this is, how do I do teratoma studies? And you need a little bit of background on this front, is that a teratoma is a benign tumor that's formed because embryonic stem cells don't have a signal to differentiate. So if you transplant them in a particular site, they grow. And the only signals they have are within the tumor mass, so they differentiate at random. And since embryonic stem cells can differentiate into multiple phenotypes, what you will see within a teratoma is maybe ectoderm, endoderm, and mesoderm. So it may be bone, cartilage with hair maybe, or you might see nervous tissue along with hair. So that's a complex tumor in that sense. But it's a benign tumor because it's self-limiting, because all the cells, once they've differentiated, then you don't repopulate, or you don't get metastasis of that tumor. So, and this happens because there aren't signals for the cells from outside regulating the process of differentiation. Normally we test for residual embryonic stem cells by transplanting in a particular site in an immune compromised mice, so that you don't reject the cells from human in terms of being able to do it. And you do about 2 to 5 million cells, okay, in terms of getting there. And the success rate generally is between 80 to 90%. So about 10% of the time, even though you've done the transplant, just because of the way the surgical process is, it doesn't work. Okay. So the problem for me is the following. It really looks like success that I've depleted all my cells, because my treatment for Parkinson's requires I use less than 1 million cells. So if I take my end product and I do a subcutaneous implantation experiment, I'll always get a negative result, right, in the time period that I look at it. But it doesn't mean that I won't in the future get a tumor, or that I don't have residual embryonic stem cells. And I don't know how to sort that, and I don't know what is the regulatory process by which I can do that. And I can't really do a large enough N, which would be the sort of knee-jerk solution, right? When you've got a low low result or a low frequency of any occurrence, do a large enough N and you'll pick it up. But I can't do 2,000 animals. I can't do that. It's just way too expensive, both in terms of animal lives and in cost, right? So in terms of doing it. So that's a big issue. So if we could figure out a 3D model, if we could figure out a way to be able to test this reliably, or we could look at equivalence, then that would be really a huge solution. I'll skip what this thing is. This is the last slide here is, can we assess differentiation and function relatively rapidly? And the answer is yes. We can use these sorts of molecular biology techniques that I just talked about. And luckily for us, when we look at dopaminergic neuron differentiation, or we look at any other lineage in terms of differentiation, my developmental biology colleagues have worked out things in a very nice, detailed way. So I just have to go to the literature, and I can really pretty much identify markers at each of these stages in this process, or any other differentiation process that we've looked at. So I have very clean markers to be able to define stages of cells. Skip all of this. And the last slide here I want to show is this, and this is just important for manufacture scalability, an issue about generalizing things. Remember, I started off by saying for MSC, I really wanted to make sure that my cells, what media that I develop, or the package that I developed, would work for all MSC-like cells. So for the ESC field, we had this new finding, right? Just 3 years ago, we had Shinya Yamanaka telling us that there is another way to get pluripotent cells, and then that was replicated by more than 150 labs within less than a year, and now everybody can make induced pluripotent stem cells, right? So, and everybody is now wanting to use their personalized stem cells, right, for some kind of therapy. So I can't develop a separate media for everything, right? So what do I do? So for me, my big hope was the same media processes and protocols that I developed for embryonic stem cells should work with iPSC cells. So my first test was, are the protocols the same? Right? I had this whole protocol for differentiation into dopaminergic neurons. I should now be able to take multiple induced pluripotent stem cell lines, and I should be able to run them through the same system, and I should get the same result. If that was true, then it might be tweaking, but that would be like with any other line that I was using, but it's not like I have to develop whole new systems. And so the quick take-home answer is that the protocols were very similar. I took 3 different lines, 2 which had been derived by a group at Hopkins and 1 which had been derived from the Scripps Institute, and I tested them. And basically the same xeno-free conditions in the package that we'd used could be used to derive iPSC, derived neural stem cells, and then I could take those and differentiate into dopaminergic neurons, which is what you're seeing at low and high power. They're stained with TH, and I apologize, it's much better on the screen, and hopefully you'll have a copy of that. And you can show that you can purify the same way and that the same markers and the same growth factors work. It was something we expected, but it was important that we show it, right? And of course, we've only shown it with 3 lines. You'd have to really do it with a much larger N. And the take home, though, was that if it's truly become an ESC-like cell using the reprogramming genes, then almost everything that we've described for an ESC protocol has worked for us so far, right? And we've now done it with more than these 3, but that's the most complete data I have right now. So we feel relatively confident that this might actually be lucky for us, right? That it's not going to be something unique, or epigenetic changes or microRNA changes, which have been shown to be different, are going to be sufficiently significant in terms of the culture and manufacture process. Okay. So overall, we have a process, and that process seems to work for ES cells and iPSC cells. So these are just some acknowledgements. So a lot of the MSc work was done by this large group of people within Life Technologies. As you heard, we collaborated with this group in Germany. We also collaborated with groups in Singapore, particularly for figuring out the ESC-derived MSc culture conditions and protocols, and identifying the key growth factor pathway from the Bioinformatics Institute. You saw some of the in vivo work with the University of Tokyo, and of course we worked with Case Western Reserve University to look at growth factor and senescence. Most of the neural stem cell and embryonic stem cell work that we did here, apart from the stem cell team, the ESC and the clinical grade derivation was done by Stem Lifeline, which is a company based out of California. And a lot of the work on dopaminergic neuron differentiation, in fact, 90% of the work was done in collaboration with Dr. Shenmin Zheng and her lab. A little bit of the work which I didn't show you here on transplant was done with Julie Anderson's lab at Shenmin, and I have a small group at the Buck Institute of Wells. I didn't show you this data, but we can take the same neural stem cells that we harvest from these clinical grade lines and make glial precursors from them, and we can use those for different kinds of therapy, and that work was being done with Johns Hopkins Institute with Dr. Maragakis and Dr. Jeff Rothstein. I'll be happy to take questions.
Thank you, Mahendra. So let's open it up for questions. Steve Stice?
Mahendra, thanks for the talk. Very, very nice. The issues around clinical aspects now with Cyrus's failed phase 3 clinical trial with MSCs, how much of that is due to biological differences, and how much of it's due to the processes of manufacturing those cells, in your opinion?
So it's a guess, given we don't have insight into all the details even. I think about 50% of it is just biological variability. It's hard to select the right kind of patients, and you made a bet that you're going to go with a single type of line, right? It's not a pooled population as well. They selected something. So that's a big issue as far as we're concerned. If you look at the data, you can't do a post hoc analysis as far as FDA regulatory approval, but you can do it for your own interest. And if you do that, you can parse out the patients, and you get a subclass which respond really well, and there's a subclass which don't respond as well, or at all. And if you could discard those patients, you'd have a very, really dramatically improved result. And unfortunately for them, that's a big problem. The problem is we can't proactively identify which ones are going to fail, right? But it's useful to remember, but this is true for breast cancer therapy as well, right? Herceptin works only in a subclass of patients. So in fact, now it looks like, one of the major drugs that we use for anticoagulation, right, works only in 10% of the patients. They've identified the gene that's important. So unfortunately for this, we don't know what the cause is. But I also think there's a process issue. They committed to certain kinds of manufacturing processes and the passage number based on what was known at that time, right? And that's the best quality that we had at that time. But they didn't optimize for the function that they wanted to keep for the first clinical indication, right? And it's important to do that. Right? So.
Barbara?
Yeah, hi. I really enjoyed your talk, and I think you hit a lot of things that really have to be addressed when we try to make a stem cell as a product. But actually, Osiris' first product is on the market. It's not regulated in this way, and as you know, they basically are selling it now through Nuvasive as a stem cell enhanced bone graft. And it's being it's regulated not by FDA but by the American Association of Tissue Banks.
Yes.
And I'm wondering, and there's anecdotal evidence that it works, and it's working pretty well, and it's selling like hotcakes. So what is different about that product from the ones you've described is that it's not pure and makes no claim at being pure. And I'm wondering if there's another product we're going to purify out the stuff that we might actually need.
Absolutely. So the reason, so the two reasons why Osiris did what it did in terms of with Nuvasive, right? If you have, the guidances from the FDA are the following, right? That if it's minimally manipulated and it's less than 72 hours in culture, that's the rough guideline, and it's being used in an autologous way, it's an analogous use to its in vivo use, right? Then what that means is that there's no FDA regulation, and then you can even market it or sell it, but it's a process, right? And so that's one reason why they're doing it. And so they had no incentive to purify, they had no incentive to do anything. In fact, it was better not to do it, right, in terms of being able to do that. The data that product works better than a purified product is actually not there, and the few studies which have suggested purification might be useful has actually suggested purification would be important. However, that enforces a whole set of regulations and a whole set of sort of different criteria in terms of what you can approve and what you can label and what you can sell it at. That didn't seem to be a good strategy for them.
No, I agree totally about the strategy, but I'm also wondering if there is a scientific lesson we can learn from that as well.
Oh, yeah, so for that, while I can't say that for MSC, I would absolutely say that's true in the nervous system, because there we have very clear data that a purified population doesn't work, but if we actually add support cells, in this case the astrocytes, then the survival rates are significantly higher, right?
So my next question after that is, if we agree that it's nice to have maybe some helper cells around, what's the immunological consequence of that?
So we don't know, but it's important to find out, because the way FDA or anybody regulates the process is the product is not just the cell you think is of benefit, but it's the composite, right, of everything that goes into that tube. And so that will be a normal question that will be asked, is what do everything else in that tube do? Whether it's the residual media, whether it's the residual DMSO, the residual growth factors, or the antibodies you use for selection, or the support cells that may be carried over. And you have to identify them. And in fact, what the FDA says is, for a consistent product, you're not just going to say, I have, for example, if I was looking at dopaminergic neurons, that you can say I have 150,000 dopaminergic neurons, but I also have to say what the other cells are, and that the relative ratio remains the same from lot to lot. So you're absolutely right. We need to know, and that becomes an important requirement.
So a number of the approaches that you showed to try and scale up production of these is leveraging existing technology. How successful do you think it's going to be, or what are the limitations of that? Because those were developed around and perhaps not quite as much of the time concerned about self-phenotype things, whereas stem cells, if they are different in a sense, have more reversibility or ability to change due to their surroundings. How much of that is that going to affect the ability to use existing technologies versus the need to have to completely innovate development?
So far, every cell type we've looked at, it's it's been possible to adapt existing technology to doing that. Okay. The adaptation sometimes has been difficult, right? But it's been possible to do. So it's not that I've had to identify any fundamentally new methodology to be able to sort, or to be able to select, or to be able to grow in suspension, for any of the things that I pointed out. So beads don't work as well. I actually had to figure out that when I made media, define them, that inhibitors of differentiation were an important component of the media, right? Before I could use it for stem cell growth. When I grew things in suspension, it turned out that there was a mass effect where cells would differentiate simply because of the microenvironment changes. But I could correct for that if I could do a size selection, so that I never let things grow beyond a certain size, right? And those could be automated, and those could be scaled up, in terms of the right way to be able to do these things. But it took time to figure those things out. But so far, it's not like I've had to say, well, I can't sort with beads at all, and it's never going to work, and I need ultrasound, or I need heat methods of selecting, or I need novel methods of suicide gene control, simply because there's no other strategy or choice available. So far.
Okay, maybe this is a good time to bring this to a close. Some of you may want to come up and talk to Dr. Rao. Also, I think there's some students for lunch. That's in room 1107. Mahendra, thank you very much again.